So, good morning, everyone. My name is Lucía Alonso. I'm a PhD student at the company In the Forest, and today I'm going to present you the work entitled Assembly of Xylem Inhabiting Bacterial Communities for Sustainable Management of Olive Vascular Diseases, which was carried out at the Institute for Sustainable Agriculture of the SIC in Córdoba. So a bit of a background, diseases caused by Verticillium daliae and Xylella fastidiosa are vascular diseases that affect a wide range of woody plants. In the case of Verticillium daliae, it's a soil-borne fungi that can infect over 70 host plants, and its mycosclerotia can remain viable in the soil for up to 14 years. Meanwhile, Xylella fastidiosa is a xylem inhabiting bacterium regulated by the European Union as a quarantine pest, and it's transmitted by insect vectors. It can affect over 700 host plants, and both diseases have in common that they colonize the xylem, and the symptoms that the plant develop are really similar and also similar to severe drought. Both of them cause high economic and environmental impact, and right now the control measures are partially effective as they are focused on eradication and containment. Therefore, there is an urgent need for integrated management to prevent the spread, reduce the incidence and the severity of these pathogens. Plant vascular diseases cause important cultural, social, and environmental effects other than the more evident economic ones. And one of the most affected species is the olive, Olea europaea, which is a key component of Mediterranean agroecosystems. Spain holds the first global producer, according to FAO, and this crop occupies over 52% of the total woody crop area in Spain, almost 16% of the crop area, and more than 5% of the total geographical area of Spain. And as Jorge just mentioned, it's a expanding species, so we expect it to cover even more during the next years. So in this challenging context, endophytes are emerging as a promising strategy because they have mechanisms related to biocontrol for these diseases. There are two types of mechanisms related with biocontrol. Direct mechanisms are related with inhibition and enzyme production, and indirect mechanisms are related with plant growth promoting traits. So we hypothesized that we could develop bacterial synthetic communities of these endophytes with mechanisms related with biocontrol to try to control these diseases. So the objectives that we set was to develop syncoms using olive xylem inhabiting bacteria with biocontrol potential against these two vascular pathogens. And to reach this goal, we developed the following workflow, starting with the isolation of a bacterial collection of over 300 strains. Then we selected the 105 representative ones to evaluate their direct and indirect biocontrol mechanisms. Then, with the 26 most promising strains, we evaluated their metabolism profile by carbon sources assimilation, biochemical sensitivity, and pathogen in vitro inhibition. And then we performed compatibility assays to finally be able to select the ones to design the syncop. So, starting with the evaluation of direct and indirect biocontrol mechanisms, we performed 18 different assays, 5 of them related with plant growth promoting mechanisms, which are the indirect ones, and the remaining 13 with the enzyme production and direct mechanisms. Most positive results, as you can see in the pictures, are shown with a transparent halo, a change of color to red or blue, bubble formation, precipitate formation, or the turn to brown color in the filter paper. Then, with the 26 most promising ones, we evaluated their carbon sources assimilation using Biolog 3 microplates, which have 71 carbon sources lyophilized into the wells and 23 biochemical sensitivity assays. And then, with those same 26 strains, we perform dual cultures with the pathogen isolated as a lung and the tested strains with droplets. And then we perform compatibility assays. The compatibility assays allowed us to discard the more incompatible ones shown in red, and taking into account these results, along with the complementarity into biocontrol mechanisms, the in vitro inhibition, we selected 5 promising strains to assemble the 2 syncoms. Also, we confirmed the syncom performance against the pathogens to be consistent with the one showed with the individual strains. And right now, we are testing the 2 syncoms that we've developed in tobacco plants as a model plant and also Olea europaea. These experiments are currently ongoing, but preliminary results look really promising. And as take-home messages, I want to highlight that we characterize a diverse collection of olive endophytes with biocontrol potential against vascular pathogens. The selected strains showed compatibility, supporting the development of effective synthetic communities, and preliminary implantar results are promising, encouraging further experimentation under field-like conditions. And overall, this work provides a foundational step towards sustainable microbiome-based strategies for olive crop protection. I want to thank the Biology and Ecology of the Soil Microbiome Group at the IASTESIC, all the funding from national, regional, and international projects, some collaborators, and the grant from Agencia Estatal de Investigación. And also, thank you all for your attention.